So it's my pleasure today to have Carla A as our Warren Seminar Lecturer. So Carla did her bachelor's and master's degree at SUNY Buffalo in chemical engineering, and her PhD at Northwestern in chemical engineering, at least according to her CV. And then obviously saw the light at some point and moved towards things environmental. And she spent a few years at ETH in Zurich as a postdoc and a research scientist. And then for just about 4 years, she's been an assistant professor at Pitt in civil and environmental engineering, and she's got an appointment in public health as well. And Carla does lots of interesting work in persistent organic chemicals, particularly fluorochemicals, which are very relevant in Minnesota. So I wanted to have Carla come speak. And with that, I will turn it over to Carla.
All right. Thank you so much, Bill. And thank you all for having me. This is going to be hopefully fun for you. We'll see. Let me share my screen and get my slides up here. And as Bill mentioned, most of my degrees, all of my degrees are actually in chemical engineering, but I did work with my advisor at Northwestern was Kimberly Gray. So I was in a civil and environmental engineering group for my PhD. It's just that she has a joint appointment in chemi, so she kind of stole me over while still allowing me to get my chemical engineering degree. So I've seen the light a bit earlier than just recently. Anyway, so today I'm going to be talking a lot about some of the work that my group is doing and that I'm doing in collaboration with other scientists on trying to solve this problem that is increasingly recognized as a pretty big problem in terms of environmental contamination by persistent chemicals, and that's PFAS. So before I start, if I can get this to work, I wanted to acknowledge my group. This is my research group at Pitt. They're a group of really great people that have helped push my research agenda along the last 4 years. And we, in general, work on chemical fate and impacts in human environment systems. We do a lot of work on PFAS, which is all I'm going to be talking about today, but we also do work with flame retardants and PAHs and some other things of interest. And this is my group right now under these weird conditions we find ourselves in 2020. But they've been able to keep up their work in the lab kind of at a reduced scale. And luckily we do a lot of modeling, and that work has been able to continue without much pause aside from the general feeling of dread that is 2020. So for those of you who are not very well versed in PFAS, I wanted to just give a little primer on what they are, what they look like, and some of the issues around them. So on the screen now are some of the quintessential representatives of the PFAS class. So PFAS are per and polyfluoroalkyl substances. This means that they are alkyl substances where the hydrogens have been replaced to some extent by fluorine. So the perfluorinated ones have all of the hydrogens on the backbone replaced with fluorines, and the polyfluoroalkyl substances still have some CH groups left in the molecules. And you'll see a lot of acronyms. They can be confusing, but in general, they tell you something about the chain length of the chemical and the head group. So for example, PFOS is perfluorooctane sulfonate. That tells us it has 8 carbons, and the S means it has a sulfonate acid group. And the PFOA is a perfluorooctanoic acid, and so again, it has 8 carbons, but in this case, it has a carboxylic acid group. So actually, PFOA has 1 fewer fluorinated carbon than PFOS does, because the 8th carbon has is part of the carboxylic group. And then you have a polyfluoroalkyl substance like an 8,2-fluorotelomere alcohol, which has a perfluorinated part and then this hydrogen substituted part. And this part is a part at which some degradation can happen. And so there are many types of polyfluorinated substances that are released into the environment, which will break down at that telomere or at that non-fluorinated site or near that non-fluorinated site. And what will happen is it'll generate a number of shorter chain perfluorinated acids. And so even trying to figure out where these PFAS come from in the environment becomes complicated because they can be transformation products of other substances. And the reason these have become so ubiquitous in the environment is because they have a lot of uses. This fluorinated carbon chain is highly surface active, and so having this very hydrophobic and very polar ends means that they make great surfactants. They make great stain and water repellents. And so this is just a little comparison table for different types of surfactants that give you an idea why the industry likes them so much. So they're highly effective and they're really, really efficient. So you need much less weight percent of PFAS than you would of, say, a hydrocarbon or a silicone surfactant to give you the same activity. And at the same time, they are incredibly resistant to breakdown. So they have excellent resistance to acids, alkaline, oxidizing media, thermal resistance. And even though they are quite a bit more expensive, all of these other properties means that they're widely used. And probably those of you who know a bit about PFAS know about them from their use as surface water and stain repellents. So they were widely used in Teflon, so nonstick cookware, etc. Scotch Guard stain protectors. Teflon also, of course, is used widely in laboratory equipment. And so if you're trying to analyze PFAS, it's going to be a huge headache because there's background sources of it everywhere. But the use that really led to a lot of widespread environmental contamination was in the so-called AFFF. And this is an aqueous film-forming foam that is used to fight hydrocarbon fuel fires. It is a very highly effective way to put out fires that are very difficult to extinguish by other means. Unfortunately, it wasn't only used to put out fires, it was also used for all sorts of training activities. And this little video just shows you some of the maybe the less well thought out ways in which it had been used in the past. And you can see here, this is in an airplane hangar where they're testing these big means to discharge foams. And normally what happens after a test like this is they would open up the hangar doors and just shovel out the foam and spray it down, and it would wind up contaminating the surrounding environment. So recognition that this was not good practice has curtailed this use of PFAS in testing like this. However, because of the persistence of PFAS, it means it is still found all over the United States and all over the world in places where this AFFF was used. So this is a map from the Environmental Working Group that shows sites of known water contamination by PFAS. And the majority of these sites are there because of the use of AFFF, although there are some other industrial areas. And one thing I always like to point out about this map is that the relative density of points in some areas do not correlate with higher use of PFAS. It tends to correlate with more investigations about PFAS. So the more you look for it, the more you find it. So for example, Michigan here has had a really strong testing program. They're covered in dots. And in addition to this AFFF, there is more recognition of some consumer-facing exposure sources that people are concerned about because they know that it leads to people having PFAS in their blood, not just from contaminated drinking water, but from things like coatings on food packaging. So pizza boxes, popcorn, especially microwave popcorn bags are known to contain PFAS. Burger wrappers, anything that might otherwise be stained by oil and that you wouldn't want to realize how greasy the food you're eating is, right? So you'd hate to pick up that cookie and realize just how much oil is in it. And so we're going to put a nice wrapper around it that prevents any of the oil from soaking through, and it looks nice and clean and not as unhealthy for you. Some lesser known sources that have come to light are things like dental floss. So-called Glide dental floss glides so nicely because it's got Teflon in it, right? So that makes it easier to insert between your teeth and move around. However, they have shown that there is a correlation between people who use this particular type of dental floss and higher PFAS levels in the blood. Things that you might not think about where PFAS wind up, like your shaving gel. So that has Teflon in it. Many makeup products and face products that help things glide smoothly onto your skin. Things that you would never consider, for example, like eyeliner. So there is a lot of PFAS that people are directly exposed to, and maybe in ways that they wouldn't necessarily think about. These, however, are still pretty visible uses, especially for things like makeup. You can usually find the ingredients for these online, if not on the actual packaging, and so you'll be able to see that they use some sort of fluoropolymer or fluorinated surfactant. But there are a ton of also hidden sources. And this was a really great work that was recently published by Juliana Gluga in ETH, where she basically dug through acres of patent literature and other literature to try to discover which PFAS are used where. And this is one of the biggest challenges anybody working in PFAS has, is that it's so pervasive in so many industries, and we don't have access to that information generally because it's often confidential business information. So she dug through a bunch of literature and especially patents and was able to find more than 200 use categories and subcategories that were linked to 1,400 individual PFAS. And as she mentions in the abstract, this is not an exhaustive study. This is basically, she had funding to do this for, X number of months, and this is how much she was able to uncover in that time. And if you can put in more time, if you have the resources to do that, you will find more uses of PFAS. And one of the concerning things here is that many of the categories were things that were not previously described in the scientific literature. So the use of PFAS in things like ammunition, climbing ropes, guitar strings, artificial turf, and as a soil remediation component. So it's really no wonder that we keep finding it everywhere in the environment, because it turns out it's just used in so many different industries. And so through the life cycle of producing these products and using these products and then disposing of these products, you wind up having widespread emissions into the environment. And as a result, you get widespread exposure to the population. So this is from the CDC. On their website, it clearly states that most people in the United States have been exposed to PFAS and have PFAS in their blood, especially PFOS and PFOA, those first two structures that I showed you. And this is a concern because these are not inert chemicals. It turns out that they are being increasingly linked to a variety of health effects, and especially they're showing that even at quite low concentrations, PFAS can be associated with things like infertility, premature birth, and low birth weight. And in fact, as part of this recent comprehensive study that came out of a CTAC meeting that tried to categorize all that's known about at least the PFAS that have been well studied, one of the most striking observations is that for the well-studied PFAS, there is just such a breadth of biological systems that are affected by these substances. So there are developmental effects, there are effects on lipid metabolism, there are effects that are associated with kidney cancer, testicular cancer, thyroid disease. There are some lower certainty but have been links to things like breast cancer, obesity, low sperm count in children that are exposed in utero, et cetera, et cetera. So in part, our increasing recognition of these issues is leading to more and lower limits for drinking water concentration. So here's a place where the federal government has really been a bit slow, I would say, in updating what we know about PFAS toxicity and what we should do about it. So the federal level of concern, it's not even an MCL. We don't have a federal MCL, maximum contaminant limit for PFAS, but the federal guideline level is still set at 70 parts per trillion, which when you compare it with other chemicals is still a small number. This is a part per trillion that we're worried about. And so when you think about it as an engineer and you have to figure out how to clean your water to get to that level, that's a big challenge. But over the years, that level has been dropping more and more. So here's the EPA level in 2010. And here are the more recent levels. So in 2000, I think this is actually 2009, the EPA was up here. In 2016, they dropped that to 70 ppt. That's the current federal PFOA guideline level. And every year since then, states have been getting lower and lower. And in 2020 now, we are down to 10 parts per trillion and below being seen as the quote unquote safe level for drinking water consumption. And that's taking into account all of the other sources of exposure that we have. And this was a paper that was just published by Gloria Post. She works at the New Jersey Department of Environmental Protection, and she is an absolute thesaurus when it comes to understanding state guidelines for PFAS and how these decisions are made. So I would, if you're interested in those water guideline levels, I would really recommend this paper. And as she mentions in there, as of May 2020, 12 US states have developed their own guidelines for PFAS in drinking water, and of these, 9 have developed drinking water guidelines that are more stringent than the EPA's level, and 10 have developed guidelines for PFAS that are not included in the EPA's level. So that's the other thing, is the EPA right now only looks at PFOA and PFOS, and there are many more PFAS than that in the environment. And here's just a small illustration of that. So this was a paper in 2016 by Leo Young and Scott Maybury that showed that humans are exposed to a whole bunch of unidentified organofluorine. That means that there's this organofluorine signal that can be found in human blood. We don't know what those chemicals are. So we can figure out the other chemicals that we have standards for. So we know which are PFAS and PFOA and their precursors, but all this stuff in black, not sure. Probably some sort of PFAS, because there are not a lot of other sources of organofluorines. Could include fluorinated drugs, which there's a huge argument about whether these should be considered PFAS or not. But in any event, they're there, they're in our blood, and we don't really know what they're doing. So there are big questions that emerge from this illustration, like which PFAS are going to have those effects that we saw for PFOA and PFAS? How are we going to be able to evaluate the health risks from this ongoing exposure to these mixtures of chemicals, many of which we don't know the structures? And how are we going to marshal our really limited resources to address this widespread contamination, right? We want a clean environment, but that has to get actually done somehow, right? So we have to have the technology, we have to have the funding, we have to be able to implement it on a wide scale. So in my group, and just as a final highlight of this, if you're interested in the zoo of PFAS substances that we do know a little bit about, the EPA has a CompTox dashboard that's meant to facilitate computational toxicology. And their current PFAS master list, which was really recently updated, has 9,252 different PFAS in it. These are not all single substances. Some of them are mixtures, but the amount that are mixtures is pretty small relative to the total. So you can just scroll through that website and see all sorts of interesting, highly fluorinated structures that have been registered in one or another database and may or may not be used at unknown quantities in different products. So it's a bit eye-opening, and it also, if you're interested in computational toxicology, it gives you a lot of stuff to play with. So what is my group trying to do about this? We are trying to come up with some bottom-up approaches that we can address this sort of iceberg of unknown PFAS and whether or not they are harmful. So the tip of the iceberg is the stuff we know well, right? So that's your PFAS, your PFOA, a handful of other substances where we have data. There's a few more that we have a little bit of data about that are known to be in consumer products. So for example, the perfluoroalkyl ethers are a subset that are getting a lot more attention now because they've been found in the environment. But we don't have too many property or effects data available for them. And then there is the whole rest of the zoo of potentially thousands of additional PFAS with no data available at all, including where they're being used and what concentrations. So what can we do about this? So if you're thinking about thousands of substances that are very persistent in the environment, a chemical by chemical approach is simply not feasible because it'll take you forever to get to it. And during that time, it will be released. And if they have a potentially problematic impact, it's going to be very poorly reversible exposure because these substances are difficult to remove from the environment and they're very persistent. So we would like to be able to use whatever existing knowledge we already have and the one fundamental piece of information we can get from these chemicals, which is their structure. So I'm going to give you a few highlights of some of the work that my group is doing. I'm not going to be able to go into a ton of detail, but I'm happy to chat further about anything that you're particularly interested in. And I'm going to start with what wound up being the first paper to come out of my PIT group when I started. And this is working with my student, Wei-Xiao Cheng, who is amazing. And she should be finishing his PhD in next semester. But he started out by putting together what we call a bottom-up PBTK model for PFOA in the male rat. So if you have a PFAS that is very well studied, a great way to look at where it goes in the body and what happens is using what are called physiologically based toxicokinetic models. If you're from the drug side of things, you talk about pharmacokinetic models. If they're unintentional things that are entering your body through the environment, we call them toxicokinetic models instead. And we use these models to integrate our understanding of uptake rates for these chemicals, tissue distribution, where do they accumulate, and elimination kinetics. So how long are they going to stick around? And the models that we build are a little bit different because they include some components that are not normally included in PBPK models, and then our approach to parameterizing them is a little bit different. So we call this bottom-up PBPK because we are trying to build models that use existing physiological and in vitro data, but do not fit the model output to any in vivo experiments. So we want to be able to make models that we can then apply to chemicals where we don't have any in vivo data. And the other component of it that is specific for PFAS is that we explicitly consider protein binding and membrane transporters when we're looking at PFAS fate. Because PFAS don't act like your typical hydrophobic neutral organic contaminant. They're not going to accumulate in the lipid tissues like a PCB would or a PBDE would. Instead, they tend to bind to proteins. In the blood, they are associated with serum albumin. In the liver, they're associated with liver fatty acid binding protein. And they also seem to be transported in and out of tissues through membrane transporters. And this is especially important in the kidney, where for chemicals like PFOA, the half-life in the organism is really dictated by the degree of reabsorption of the compound with these kidney transporters. And that leads to really interesting behavior because these transporters have sex-dependent expression patterns. And so you'll have higher half-lives in males than you do in females because the females don't reabsorb it as efficiently. So it's kind of a fascinating scientific area to study. So we try to put all of this information together, and we need to find data for things that other people have not gathered data for necessarily, or were not thinking about your model when they did it. So I like to put in this slide here just to give students an idea of how we try to find data for parameterizing models like this. And I like to talk about the engineer's approach versus the bench scientist approach. I don't know if this is really the engineer's approach. It's my approach when I started working on PFAS. So this is my the first paper that I published in PFAS in 2013. And I kind of like it just because of the sheer audacity of trying to model a fish tissue like plumbing. But I'm an engineer, so that's what I do. So I was trying to figure out what the surface area for blood tissue exchange was, for example. And so you model the capillaries as tubes with a certain radius and a certain thickness, and then you figure out what these surface areas are with really sort of back of the envelope engineering calculations, knowing physiological rates like the blood volume and the blood volumetric flow rate in different tissues. And, you know, it worked out okay for that particular study. But if you have real measured data, I might suggest you try that instead. And a bench scientist, we use a lot of data that come from in vitro studies. So for example, when we're talking about these transporter studies, nobody had really studied mechanistically how these transporters were interacting with PFAS. They just know whether the transport happens faster or slower when the transporters are present. So this was a study that was done, I think this is from Weaver et al., where they actually looked at cells that were expressing certain transporter proteins, in this case the OAT3 protein that looked at PFOA uptake, and cells that were not expressing the transporter. And so we can basically break those curves up into your total uptake, your net, your passive diffusion, which is what happens when you don't have the transporters, and your net active uptake, which must be due to the transporter. So this is how we parameterize that part of the model. And what's great about this too is that it really clearly shows that while passive diffusion of PFAS can happen in cells, it is much slower than what happens if there are transporters present that facilitate that uptake. So that to us clearly showed the importance of including of transporters in models. So putting all that data together, we came up with a model that had 72 independent parameters. And then we wanted to use those parameters to predict to compare with 7 data sets from 3 studies that had both high and low doses of exposure, oral and IV, plasma time course, and tissue distribution. And so this is what the outcome of that looked like. So in the dark black line is the geometric mean of our model predictions. And then the different colored symbols are the different studies that we tried to replicate. So in the top, we have a high oral dose, a high IV dose, and then at the bottom, we have a low oral dose and a low IV dose. And you can see that the model does pretty well, definitely within the 95% confidence interval, but even close to the geometric mean, even though we didn't fit a single parameter. And we use the same parameters for the different studies, we just changed the exposure settings. So if you have enough data, you can come up with a well-parameterized model that reproduces in vivo behavior for different doses and exposure routes, which is a great way to do in silico testing for new chemicals. And they do this sort of thing for drugs all the time. But unfortunately, the data that we needed to parameterize this model, for example, those uptake rates for different transporters, is totally unavailable for the majority of PFAS. Moreover, it's totally unavailable for most organisms. And it turns out that because PFAS has these interactions with different proteins and transporters and receptors, it also shows really large species-specific differences depending on how different those proteins are across different species. So if you don't have the right model organism, you may also not be replicating the information you need. So having done that first proof of concept for PBTK, we thought, well, okay, how are we going to parameterize for chemicals where we don't have these data? And so we started thinking about new ways to be able to predict passive diffusion, membrane transporter interactions, and then binding to proteins both inside and outside of the cell. And so that brought us to considering molecular modeling. And so we started working with molecular dynamics as a way to both predict these interactions and to provide a means of screening across different PFAS to see which PFAS would tend to have the bigger interactions. So the only things you need to be able to do molecular dynamics are a high quality protein structure, which you can get from the protein data bank, or you can actually model it if you know the gene sequence using something like the FIRE2 server. And you need a structure for your PFAS, which you can draw, or it might be available for a few of them in the protein data bank. We normally use Avogadro, partly because it was created here at Pitt, but also because it's free and easily available. And then you take those two structures and you create a structure of the docked compound using molecular docking. And then you take that compound and you do a calculation to figure out what is the free energy of binding. So that's what this part is here. This is the most computationally expensive piece because especially the entropy part can be quite difficult to model for a large protein. And so the larger your protein, the more computationally expensive it is. But the nice thing about it is that it breaks down this free energy of binding into the different components that drive that binding. And so you get to learn whether it's the electrostatic interaction, if it's the hydrophobic interaction, what is it that's making such a strong interaction between your protein and your structure. And just so you'll see in the next slides where I talk about the results, if you have a lower delta G, it means you have stronger binding. So more negative means stronger binding. And if you assume that PFAS binding to certain proteins like this liver fatty acid binding protein I have up here, or serum albumin is a major driver for bioaccumulation, you can use these interactions not just to parameterize PBTK models, but actually as a screen to say, if I run a bunch of different PFAS structures through this framework, and I see which ones I rank them by the strength of interaction, the ones with the strongest interactions are likely to be the most bioaccumulative substances. So it helps you do screening in that way as well. So this is what we did. We wanted to look at both well-known chemicals like your PFAS and your PFOA, but also some of these emerging substances, these ether PFAS that had not yet been very well studied, and predict what we would think would be their bioaccumulative potential. So we did this with molecular dynamics, and what you get is basically a predicted delta G, which you can compare to your measured delta G. And so I just wanted to point out a couple of things here. As I mentioned before, as the delta G gets lower, the binding gets strongest. So the bottom left corner of these plots are the ones that bind the strongest to liver fatty acid binding protein. Also that the numbers on the predicted delta G are lower than the numbers on the measured delta G. So that is just to emphasize that these predicted values are relative. They tell you there's good correlation, right? So our correlation coefficients are high, but you need to have some data to pin it to in order to get an actual number for what you predict it to be, because we'll tend to over-predict the binding strength with these approaches. But they will let you rank things successfully. And here are two of these emerging substances that we were able to evaluate. GenX, which is one that is known to be especially prevalent in North Carolina because of a fluoropolymer manufacturing facility there, and F53B, which is widely used in China as a replacement for PFAS and has recently also been detected in the US. And you can see that F53B is as or more accumulative or binds as strongly as PFAS. And this is what we're finding. We're finding F53B accumulating in fish tissues to similar degrees as PFAS. Whereas GenX is actually less bioaccumulative than PFOA, and that's due really to its short chain length and branching. And then you can use your simulations also to figure out what are, for example, the key amino acid residues that are interacting with those PFAS that are leading to that interaction. And this is a great way to think about how to use these models, not just for evaluation, but also for design. How can I change the structure of my ligand to disrupt some of that binding and maybe make it less bioaccumulative. So the modeling workflow is great for predicting relative binding for both legacy and emerging PFAS, and it gives you some insight into why that binding occurs. So this was some work that we did specifically for liver fatty acid binding protein. It's a nice protein to model because it's small. It has one well-defined binding pocket. So we were able to do that with molecular dynamics pretty well. Moving on to larger proteins and more complex proteins becomes challenging because it becomes much more computationally expensive to do. So even though it's modeling, you're still talking about weeks to get results back for one protein and several ligands. And we actually want to move to a system that is even more complex because we're really interested in looking at transporter activity. Because transporter activity is going to be key to figuring out toxicokinetics in some really important tissues, like the gut and the brain. And there are more and more emerging studies that make us think that we should be worried about things like PFAS crossing into the brain. So for example, this study in 2013 looked at cadavers and found really high concentrations of the C6 carboxylic acid, PFHXA, in the liver and the brain. And there have been studies that looked at cerebrospinal fluid in people and comparing that to serum and finding that there is a different distribution in CSF than in serum that shows that there's some sort of discrimination happening. And we expect there to be discrimination because the blood-brain barrier is meant to keep things from accumulating in the brain that shouldn't be there. But we find some alarming amounts of very long-chain PFAS that are not present as high relatively in the blood that tend to accumulate in the brain. So for example, if you see these two little slivers here, this light blue one is perfluorotridecanoic acid. So there's a C13 carboxylic acid, which seems to be overrepresented in cerebrospinal fluid. And we see a similar thing for ratios of PFAS between amniotic fluid and maternal blood, that there seems to be some discrimination where you find actually higher levels of the short chain compounds. And so understanding transporters will help us understand all of these dynamics better and predict which PFAS we should be concerned about. And it can help us answer questions like whether moving to short chain PFAS is safer or what drives discrimination crossing both the blood-brain barrier and the maternal fetal interfaces. So I have a new student to the group who did a summer project with me who was really interested in looking at brain transporters. And one of the things that's driven my work on PFAS for a long time is the similarities between PFAS and saturated fatty acids. And I think that that is behind a lot of why they wind up where they do and they have the interactions that they have. And in the brain, there's a lot of fatty acids that have to come in from the blood. And so it's possible that the PFAS are using the same mechanisms to accumulate in the brain that the brain is using to bring in needed fatty acids. And so Cara wanted to look at the PGP, P-glycoprotein transporter in particular, and use molecular modeling to try and figure out whether PFAS would be a substrate or would suppress the activity of PGP. But there's a lot of challenges in trying to model transporters. So transporters, unlike the proteins that we've been looking at before, are present within the membrane. So they interact with the membrane, and they tend to have both, outside membrane domains and transmembrane domains. And often they change their conformation in order to affect the transport that they make. So here, for example, are two examples of substrate binding protein that have inward-facing and outward-facing domains, depending on where their substrate is moving into. So they're basically shuttling things in and out of cells all the time. And unfortunately, because they are difficult to model, they're also difficult to crystallize, transporter structures are really underrepresented in the protein data bank. And the ones that are there tend to have been crystallized in one or other conformation, so you only have a snapshot of what they look like, right? You won't have the full picture during their activity, and these conformation changes are required for their function. So without being able to model that, you're missing a big piece. And that's true for PGP, which is the protein we were trying to look at. So in the available experimental crystal structures for PGP, we only found two, one for human, one for mouse. They were both pretty good quality, so they had enough resolution that we could use them for molecular modeling, but they were in different conformations. So the human PGP is only available in the outward-facing conformation, and the mouse PGP was only available in the inward-facing conformation. So we're kind of getting two halves of the pictures but from 2 different species. And so we wanted to see basically what can we get from this, given the limitations that we have, especially since there were some in vitro experiments that indicated that both PFOA and PFAS could be inhibitors to PGP function. And if that's the case, then that would mean that one of the major efflux transporters of the brain might not be operating properly because of the presence of PFAS and PFOA in circulation. So what she did is picked up a set of PFAS to evaluate to see whether they would act as inhibitors, at least as far as we could tell. And to figure that out, we also wanted to compare these different PFAS to known inhibitors of PGP. So we looked at a first generation inhibitor, a second generation inhibitor, and a third generation inhibitor. And then we also looked at a couple of substrates for PFAS. And we used molecular docking to basically look at 2 things: the binding affinity over multiple docking simulations to see how strongly they interact with PGP, and the binding location. So can the location of binding tell us something about what their function is? And for the location, we basically used our docking simulations to find the midpoint of the coordinates of each ligand as it was bound. And then we classified that location using k-means clustering to see if we found things clustering in similar areas that had similar function. So this is the results for the binding affinity, and a couple of things that I think are worth pointing out here. This is the human PGP, this is the mouse PGP. They're quite similar. The human PGP tends to have slightly stronger binding, and it's a little bit more, a little less variability in the predicted binding. But overall, the pictures look similar. You see a strong increase in binding affinity with chain length for the carboxylic acids and also with the sulfonic acids. And you see a really striking difference between the first and second generation inhibitors and the third generation inhibitor. So this is showing that pharmacology is working. So the newest inhibitor is much, much more higher affinity. So you need lower doses of it to do what it needs to do. And that the substrates also have relatively high affinity. And so the thing that also stuck out to us is that the very long chain PFAS, which are the ones that we found were higher in cerebral spinal fluid, also have similar binding affinities to this third generation inhibitor. So they are similar in potency, let's say, with interacting with PGP. What this picture doesn't tell you is whether they're binding at the same binding site, right? So if it's the same binding site and the same binding affinity, then you can be confident that they're going to have a similar impact. So this is what we also looked at with our clustering, and you can see actually the evolution of inhibitors for this transporter, that the early ones kind of bind all over the place. And as you get to the third generation, you find that the binding is much more specific. So we ran 100 simulations for each one and then figured out where most of the binding happened. And you can see that there are clusters of, okay, it's mostly binding in these areas at the different conformations of PGP, whereas here they're kind of all over the place. So you can see the specificity increasing. And we're able to also see this with PFOA and PFAS. And we find particularly in the mouse conformation of the PGP that the PFOA and PFAS are both really well situated in the same binding area as the third generation inhibitor, not so much for the human PGP in this conformation. So here's where we find that they are acting most similarly to the inhibitor. So for us, it seems that the mechanism here would be most similar. The other thing that's interesting is if we look at the legacy and the emerging PFAS, we find very similar binding affinities and binding locations. So for these pairs that we picked out, this is PFDA, so it's the 10 carbon carboxylic acid, and this new F53B, we find both their binding areas and their binding affinities are very close. And the same thing with the PFHXS and Nafion byproduct 2, which is another one of these emerging ether PFAS. So this tells us that the new PFAS are certainly no better when it comes to interactions with PGP. So this was a bit of a whirlwind tour of some of our modeling. But basically, we can see from this that at least our model suggests that emerging PFAS are not safe alternatives to legacy PFAS, and major mechanisms of biological interaction are really similar. And there are structure property implications here. This is something that in chemicals assessment we often refer to as the lock-in problem. You're looking for a chemical that fulfills a certain function. And it turns out that the chemical structure leads it to have that function. And so you try to find an alternative for that chemical that has similar function often it winds up also having similar structure. And so you wind up going through this cycle of replacing chemicals and having regrettable substitutions, just like we've seen before with BPA and BPS. So by using modeling strategies like some of the ones that I've shown so far, you can increase throughput, but this is not a panacea because you still need data. You need data for training, validation, evaluation of your models. Luckily, some of that data can be found through other means. These are what I call incidental or complementary data. So one thing I didn't have a chance to talk about today, but is another area we're developing, is the use of machine learning to predict bioactivity of PFAS. And for that work, we actually found a lot of fluorinated chemicals in the pharmacological data, so pharmaceutical bioactivity data that were available online that we could use for training our models, and then we could test them on industrial PFAS to see which ones would be bioactive. So there may be more data out there than you think there are if you get creative about finding sources. So that was a bit of the bottom-up modeling stuff that we do, but I did want to take a few minutes to talk about some of the policy frameworks and strategies that we are taking to address the problem from both ends. Because even with these model strategies I've been talking about, PFAS evaluation is not going to solve the problem, right? The problem with PFAS is they're extremely persistent and they've already been released in a lot of places. They're still used in a ton of products. And so a lot of the goals that I have as a scientist who is interested in policy is how do we turn off the tap on chemicals like this? So I've been collaborating with a team of scientists and policy makers to design frameworks that can help in making sound policy for PFAS. And I'm just going to highlight two of them today. One is the essential use concept. This is a paper that came out, I guess now it's 2 years ago, I think, that has been taken up by a lot, which is great. So this was led by Ian Cousins in Sweden, and it's basically trying to find ways to figure out which are the low-hanging fruit of PFAS that can be immediately phased out of products without a lot of loss. So essential use is defined as something that is necessary for the health, safety, or critical to the functioning of society, and for which there are no available technically and economically feasible alternatives. So it has to both be needed, and there can't be something else that can easily replace it. And it's really based on the Montreal Protocol, which was used to phase out ozone-depleting CFCs, except for certain essential uses, and was a really successful use of policy to phase out problematic chemicals. So we defined 3 categories for essential use to aid and phase out. These are non-essential uses that you don't really need them, and it's really driven by market opportunity for the use of substances. So examples of that would be the use of PFAS, I think, in dental floss. You can have dental floss without PFAS in it. Water repellent surfer shorts. If you're going surfing, it's probably okay if your shorts get wet. You don't need them to have a PFAS coating on them, right? The second is substitutable, which is another one that is sort of easy to think about. These are uses that have come to be regarded as essential. They do have an important function, but there are alternatives that have been developed with equivalent functionality and adequate performance. And I think adequate performance is important. You need to have the performance that you need. You don't need to have the super chemical, right? Because that's what drives us into a lot of these issues. So examples here, the first one is still a little bit controversial, but I think we're moving in the right direction there, is AFFF. So this firefighting foam. There are many municipalities and entire countries that have moved away from using PFAS and firefighting foams, and they can function. Also many textile applications, so ski jackets, surfer shorts, etc. There are other alternatives that are available. The third one is the most difficult, and that is the currently essential function. These are uses that are considered essential because they protect health and safety, for example, and there have not yet been alternatives established. So certain medical devices use a coating to make them clean and biocompatible. Occupational protective clothing. So for example, there are surgery gowns that are coated in PFAS, which allows them to be both breathable and repel bodily fluids. So we don't have a good alternative for that yet, but there's a caveat here that this essentiality should not be considered permanent, and this is where we really have to drive innovation pressure to find good substitutes for these substances. And getting beyond essential use, I think essential use is really important when you have PFAS that are already being used in a product and you'd like to figure out what to do about that product. But the other thing is, should you consider using PFAS if you're not yet using PFAS in a product? And there, our response is a very strong no. And it's not because PFAS might be toxic or some PFAS bioaccumulate. It's really driven by the fact that essentially all PFAS are extremely persistent in the environment. And persistence by itself has to be a concern for a synthetic chemical that is not naturally around. So people try to argue about this with sand and other things, but PFAS is not sand, right? It's an organofluorine compound that is generally not present in the environment. And the key point is that you shouldn't need evidence of an effect for such an extremely persistent chemical, because if it's persistent enough, it's going to eventually reach some environment, some organism that's sensitive. And if you at that point find out that it's a problem, it's going to be too late, because reversibility of exposure for highly persistent chemicals is really difficult. And so that also brings me to the last little piece, which is about considering PFAS and really all chemicals in a life cycle approach and how important that is. And I think that the biggest example of that, or one of the most telling examples, is the story of this dairy farmer from Maine. You may have read stories about him, who had to basically get rid of all his dairy cattle and his business shut down because he once spread sludge from a paper mill on his field 15 to 20 years ago. And the PFAS stuck around so long that then the fodder that was raised in that soil had enough high enough levels of PFAS that his cows became contaminated and he couldn't sell his milk anymore. And so that has all sorts of implications for waste disposal and compostability of products that contain PFAS in it. And just tell us that, decades down the road, we're still dealing with this problem, and it's going to be a very high cost to reduce the impacts of PFAS in cases like this. So I apologize for the technical issues earlier, and hopefully this wasn't too much of a whirlwind tour through the work in my group, but I'd be happy to take any questions you have in the no time we have left.
So people can go ahead and unmute themselves if you have a question, or if you'd like to type it into the chat box, that's acceptable too.
So Carla, I have one for you.
Yeah.
With all the docking and modeling studies that you've done, do you think it's possible to design a fluorinated chemical that has utility but isn't toxicologically or environmentally concerning?
No, because of my last point about the persistence being bad enough, because we can't possibly predict all the potential things, right? So we can dock with the receptors we know about, but there might be a microorganism somewhere or some other organism somewhere that has a receptor we don't know about that it turns out to be a problem. And there's not just biological issues. That's why I think the Montreal Protocol is such a good example of looking at essential use, because CFCs were seen as completely inert, right? Here's a safe chemical that'll never be a problem, and we never thought about the ozone hole, right, or interactions with ozone. So I think because of that, it's the persistence itself. Now, what I think would be super interesting is to try to use the docking to design ways to break down PFAS. So I think there are people that are thinking about enzyme engineering and things like this to get at that CF bond, but it's going to be really hard, I think.
Carla, I've got a question. This is Paige.
Hey.
Excellent talk. I really enjoyed it. I wondered if you were getting any traction at all on your policy-related papers at the policymaker level, and what your plan was. This is a long question, but what your plan was to try to get that traction.
Mm-hmm. Yeah, thanks for that question. It's actually, so yes, in Europe. What else is new? But it's really exciting. For those who have an eye on what Europe is doing, they just released their the EU chemical strategy last week, I think. And they cited the piece efficient paper, the essentiality paper, the grouping paper, all of these things are cited in there. And they are planning a total phase out of all non-essential uses of PFAS in Europe as policy, which is awesome.
That's super exciting.
Yeah. So that's really exciting. Getting that movement in the U.S., let's talk in a couple of weeks. I don't know. We work with scientists from the EPA and from NIH, and half the time they have to remove their names from our papers because they're not allowed to say anything about policy. So it's been challenging. Yeah.
Hi, Carla. was great to
Boya.
see your talk. I really enjoyed it. I wonder, I have two kind of separate questions, and one is about the protein part.
Mhm.
I'm curious, have you, for the binding part, they seem, a bit random, and they're moving towards the direction of more concentrated binding paths, and I wonder for the binding sites, either on the lipo like protein or the transporters, is there a way we can extract the protein binding site information to actually think about design using proteins to design means to remove PFAS?
Yeah.
For example, like their hydrophobicity,
Mhm.
amino acid has charges. And I know structure is a little hard to say, but any molecular descriptors that you kind of see in those proteins.
Yeah, this is something we're actually actively working on, is we're trying to figure out how we can use proteins to design better sorbents for short chain PFAS, because the short chain PFAS are the problem, right? A long chain PFAS will stick to anything.
Yeah.
Well, not to anything, but even carbon, if you use enough carbon, we'll get the long chain out. The short chain ones are really challenging. And so we're thinking about, things like the length of the hydrophobic binding pocket and how you align the amino acids at the entrance to the pocket so that it, catches the electrostatic interaction. The issue with the short chains is difficult because they're so water soluble. And so if it's happy in water, it's hard to tell it, to get it to prefer to be on a protein. But it is something we're thinking about, and maybe it would be more like adding amino acid type functionality to a membrane or something that would help those electrostatic interactions, just align things well to capture the PFAS. And there are a few people working in that area. The problem with proteins is that they're finicky. So if you do something computationally and then you want to actually show it, they're hard to produce. They're expensive, and so to convince an agency that they should go down that route, I think, can be difficult.
Mm-hmm.
But I think there's some stuff to be learned there that you can maybe then apply to simpler materials. Yeah.
Yeah. So I guess do you think like a positively charged protein would be ideal for enhanced electrostatic? That sounds like
For the anions, sure, but I mean, when you look at so one of the things about it that's cool is you can think about multifunctional materials, right? Because if you want to get all the PFAS out, there's positively zwitterionic, non-charged. There's all sorts of stuff in there. But probably in thinking about regulation, the short chain acids are going to be the next ones to need to be looked at in water. So that's probably you're going to be looking at positively charged things.
Thanks, Carla. It was excellent.
Thanks.